welcome to OncoLive interview session. It's my pleasure to introduce our guest today, Sarah Hein, founder and CEO of the March Biosciences. Thank you so much for joining us, Sarah.
Thank you so much for having me. Really appreciate the opportunity.
It's a pleasure for us. Today, we will exploring innovative clinical research surrounding the MB-105, pioneering the targeted CAR T-cell therapy for lymphoma. We will also discuss the mechanism of action, early promising results, and how MB-105 address the key challenge in treating T-cell malignancies. Could we start with some information about your background, your professional journey, and the mission of the March Biosciences?
Absolutely. So my background was originally as a PhD scientist. That took me to Houston, Texas, where I received my training in molecular biology. But during that training, I became very interested in the process by which that bench science becomes patient impacting treatments. And that led me to being plugged into the startup ecosystem and then helping launch a company called Courier Therapeutics with Mercury Fund. Courier was a retargeted IL-2, so still a immunotherapy. And then we ended up selling that company to Valo Health a few years later. I then went to TMC Innovation to help launch a accelerator focused specifically on cancer therapeutics. And it was through that accelerator that I met Drs. Max Momokin and Malcolm Brenner and saw an earlier cut of the data that became the lead asset for March Biosciences. March Biosciences is an autologous cell therapy company. Our lead asset, MB-105, is targeting T-cell lymphoma right now in phase 2 trials, but the phase 1 trial targeted both T-cell lymphoma and leukemia. And we will expand back into those indications as well as a couple B-cell malignancies that are targeting or express CD5 as well. That would be your mantle cell lymphomas and your chronic lymphocytic leukemia. Really exciting phase 1 data. That's what led us to start this company. And really our mission at March Bio is to continue to both develop first-in-class, best-in-class therapies against new and interesting targets. And so we have a long road ahead of us to achieving that mission.
Could you provide a detailed overview of the mechanism of action for MB-105, explaining how it specifically addresses the challenge associated with the CD5 targeted CAR T-cell therapies?
Yeah, as I mentioned, MB-105 is a CAR T-cell therapy, so it's a chimeric antigen receptor, in this case targeting CD5, that allows us to really specifically direct those immune cells against that target on the tumors. CD5 is a very interesting target for this indication because it is expressed on about 98% of your normal T cells, but as a consequence of that, it's also expressed on about 70 to 80% of those T cell lymphomas, about 80 to 90% actually of T cell leukemias. And so you have an opportunity to broadly impact the patients with this disease if you can solve that engineering problem for targeting normal T cells. Now, what's very interesting about our specific CAR T cell therapy is that we can manufacture it without a lot of additional manipulations of those normal T cells when we make the CAR T cells. And the reason for that is the CAR design itself actually leverages some very unique biology where the cells end up self-removing that target, become completely negative, and we're able to manufacture that product in a very efficient way and return it to patients more quickly and with healthier cells. And so that's what was put into that phase 1 trial where we saw multiple complete responses and long-term survivors in that initial patient population and led us to being excited to drive this forward into a phase 2 study.
MB-105 has shown promising phase 1 results in relapsed or refractory CD5 T-cell lymphomas. Could you elaborate on the key finding from this trial progression in the phase 2?
Absolutely. So as mentioned, this product was actually tested in an academic phase 1 trial through the Cell and Gene Therapy Center that spans Baylor College of Medicine, Houston Methodist, and Texas Children's Hospital. And what that allowed is a really interesting partnership with those public institutions during that phase 1 to test the product, but also test improvements to the product as well through manufacturing improvements. And during that phase 1, the published results have really primarily focused on T-cell lymphoma, which is what we're carrying into our phase 2 trial. And what we saw was the original published results was only 3 patients, but actually 4 patients out of 11 were able to achieve a complete remission. And 2 of those patients are still alive today, multiple years later, coming up on 6 years. And so really demonstrating potential for long-term durable responses in an indication where, just to give you a little bit of a perspective, the last approval in T-cell lymphoma that was a novel therapeutic labeled for this indication was approved on a 9% complete remission rate and only a durability of about 6 months. And so you can see we're already in these phase 1 studies, even though the numbers are still small, beating what's the only approval in over a decade, which happened in 2024. So we're really excited about the results. Our clinicians are very excited about the results. The safety has looked quite favorable. Some of your classic sort of cell therapy side effects, such as cytokine release syndrome or neurotoxicities, have actually been quite limited thus far. And so that's been very encouraging for the overall potential of the product.
Given an cue card design of the MB-105, how exactly does this approach selectively target malignant CD5-positive cell while preserving normal T-cell functions?
As I mentioned, we manufacture our product from normal T cells, and one of the unique biology components of our specific CAR design is it actually folds over once it's expressed on the CAR T cells. And it really has to do with the binding domain that we're using, the specific hinge, the co-stimulators, and even the way that we're expressing in the cells. But when you line all those factors up, you see a very efficient downregulation, removal of CD5 from the T cells that allows us to make the CAR T cells. Now, what's interesting, and Max Mamokin has recently published this, is that this actually happens in trans as well. So from your CAR T cells to your normal T cells, they appear to be a little bit more resistant to that killing engagement and are also able to downregulate and eliminate the CD5 receptor from themselves, while we're still able to actually quite potently target and kill the tumor cells. Over the longer term, what you also see is the expansion of a polyclonal CD5 negative T cell population that does appear to also continue to provide ongoing immunity. And so we're able to maintain that balance even during the treatment of a T cell targeted therapy.
MB-105 recently received FDA orphan drug designation. How does this impact your clinical development strategy and timeline?
Orphan drug designation was a really, I think, promising or powerful support or indicator of, the fact that this is a high need indication. T-cell lymphoma is only about 15% of the larger lymphoma patient population that unfortunately is diagnosed every year. So it really is a true orphan, and sometimes supporting drug development in these smaller indications has been historically a little bit more challenging. And so by going to FDA and achieving that recognition, what that allows us to do is build confidence that this product has demonstrated promising indications of the potential benefit for survival and safety to these patients in a high need, smaller patient population. And there are some attached benefits to the orphan drug designation that FDA does provide to help incentivize development, such as market exclusivity down the road if you achieve approval of the therapeutic.
T-cell lymphoma, MB-105, is also under consideration for other CD5 positive cancer. Could you share insight into these additional therapeutic targets and any ongoing or planned studies?
Yeah, as mentioned, see, there are a couple different targets you could potentially target for T-cell lymphoma and leukemia, these T-cell malignancies. We think the data supports that CD5 is the broadest and best target for these indications, but it does have a pretty unique additional sort of component in that it actually, as a native receptor, is expressed on a small subset of B cells as well, activated B cells. And as a consequence of that, there are a couple B cell malignancies that are actually characterized by their expression of CD5. And this is exclusive to CD5 as a target versus other targets that you could look at for T cell malignancies. And so that's going to be some of your B non-Hodgkin lymphoma indications, such as, in particular, mantle cell lymphoma, chronic lymphocytic leukemia. And so there is an opportunity to impact a broader patient population. Where we have clinical data today from that phase 1 trial is in T-cell lymphoma and actually T-cell leukemia. T-cell leukemia and lymphoblastic lymphoma is a little bit more of an aggressive disease, particularly once you enter the relapsed refractory setting. It can span both adult and pediatric populations, but unlike T-cell lymphoma, which is primarily a disease of aging, does more associate with the pediatric patient population. So that'll probably be our next expansion point, but then we're also looking over the next year or two to expand back into those B-cell malignancies as well.
Given the challenge of the treating T-cell malignancies, how do you foresee the therapeutic landscape evolving, and where does the March Bio intend to position MB-105 within this context?
So if you look at our trial page right now, what you may notice is quite unusually for a investigational drug, we're actually in phase 2 trials at second line or above in peripheral T-cell lymphoma or third line or above in cutaneous T-cell lymphoma. And the reason for that, the reason that we are able to enter the clinical trial at a relatively early line of therapy is there is really no true standard of care once these patients become relapsed or refractory. And that is why they have such a high mortality rate and poor survival over even just a couple of years. And so we anticipate that, success in this indication would look like replicating or even potentially improving the response rates and durability that we saw in the phase 1 trial, and that would allow us to not just get an approval, but frankly, be the most meaningful improvement in this indication in quite a long time, well over a decade. And likely, as a consequence, our hope is that we would become a standard of care quite early on for these patients.
In the final question, looking forward, what are the next critical milestones for the March Bio beyond the phase 2 trial, and are there any plan for the partnership or expansion in your plans?
Absolutely. Right now, phase 2 trial is open. We've opened multiple sites across the U.S. and are targeting national coverage within the continental U.S. for this phase 2 trial. That's going to allow us to access these patients and push quite aggressively this year. We're expecting to have our first public data drop for the phase 2 trial towards the end of this year. I think that would be a pretty significant milestone. And then we'll look to finish this trial, the phase 2 trial as it's currently planned, sometime in late 2026, maybe early 27, depending on recruitment. And through there, as we get the data in and continue to build on the momentum that we have already, obviously accessing additional markets becomes very important. We're already looking at opportunities overseas, in particular, starting in Europe, but we have actually had some engagements in Latin America as well with some very interested groups. And so I think there's a lot of possibilities to do that, and we could even perhaps more rapidly move into those additional markets if we stage the way that we maybe not fully move our manufacturing, but open up, initially ship patient materials back and forth, and then ultimately open up additional regional manufacturing sites. So all options are on the table. We're still quite early days to completely firm up those plans for expansion, but we're definitely interested because we think we have the best opportunity for these patients to see a meaningful improvement.
Thank you very much for joining us today for all these detailed answers. I have finished my question. I don't know, is there anything else you would like to add or highlight from this discussion?
Honestly, the response to our trial opening has been not overwhelming, but very gratifying to see, both from clinicians, from potential business partners, pharma partners. I think there's a lot of excitement for the opportunities to see some meaningful improvements for these patients. And so, my ask is always quite simple, which is if you have ways, if you want to join this mission, we make ourselves relatively easy to find. Please feel free to reach out. It's going to take a real community of people, investors, pharma partners to move this forward and get it to patients over the next year or two.
Thank you.
Thank you.